Hello and welcome to Anatomy and Physiology at Glen Oaks Community College. I'm Dr. Ren Hartung. For this video, I wanted to talk about clonal selection theory, or what clonal selection theory is, and then go through the details of the clonal selection theory specifically for B cells, a type of lymphocyte. But first, more broadly, why should we have any clue about clonal selection theory? Why should it matter to us? This is a major thing for understanding how it is that the immune system works. Humans have known for a very long time, in very ancient times, we knew that once someone was infected with smallpox, for instance, they were not going to be infected by smallpox again, or at least not right away. It would take years at least for them to become susceptible to smallpox again. This understanding was so deep that even in ancient areas in India and in Africa, inoculation was begun. And the first inoculations, as I understand them, were that you would take some of the scab material from smallpox lesions, turn that into a powder, and then for people who hadn't been infected with smallpox yet, you had them snort this powder. And they would catch smallpox, but it wouldn't be as bad as if they had caught it naturally. They were more likely to recover, and they were not going to get smallpox again. So we knew that once you've been infected by something, you were unlikely to get it again, unlikely to catch the same virus again, for example. But how does that work? How is it that your body develops a type of memory to defend itself against specific pathogens? Clonal selection theory goes a good distance in helping us understand how that is. Broadly, here's what clonal selection theory is. It's basically the selection of specific white blood cells and these white blood cells are lymphocytes. Lymphocytes come in different varieties. We have B lymphocytes, that's the one I'm going to talk about in detail, but we also have T lymphocytes, and T lymphocytes consist of T killer cells or cytotoxic T cells and T helper cells, also known as helper T cells or Tregs, because they help to regulate the immune system. But it's a selection of specific lymphocytes. Here's what I mean by that. As your immune system develops, billions of lymphocytes go through development in your red bone marrow, those are the B lymphocytes, and billions of lymphocytes go through development in a gland called the thymus, which is in your chest. As they go through that development, part of the development is a random rearrangement of part of the genes for these lymphocytes. That random rearrangement makes it so that these billions of different lymphocytes can recognize billions of different antigens. If you don't know what an antigen is yet, here's the quick version. An antigen is a small particle, usually a protein or a glycoprotein, that is capable of stimulating an immune response. Antigens are the parts of the virus or the parts of the bacteria, the parts of the virus or the parts of the bacteria that your immune system tunes into and attacks so that you don't get infected again. So specific lymphocytes, those lymphocytes that happen to be able to recognize the alien antigen that we're trying to defend against, those lymphocytes are selected, and then those lymphocytes reproduce, and then we end up with defenses against those specific antigens for that specific alien. That gives us immunological memory so that we are ready to defend ourselves against this alien antigen for a long, long time, decades even. So understanding clonal selection is very basic to understanding how the immune system works and how it is that we can be immune to a specific alien virus or alien bacteria for years and years to come. The basics of clonal selection. The first part is literally the selection. How is it that this specific lymphocyte that happens to react to this alien, how is it that that cell is selected? And then the second thing that happens is for that cell that is selected, mitosis happens. I hope you remember mitosis means the division of cells. These lymphocytes that are selected for, all of the cells that they give rise to when they divide, they will have the same genetic makeup in terms of what alien it is that they're going to attack. So now you have, instead of 1 or 2 lymphocytes that can attack this alien, now you have thousands, hundreds of thousands, millions of these lymphocytes that can go and help attack this alien. And of course, the last step is the attack. How is it that that attack happens? How is it that your immune system actually goes after and gets rid of this alien antigen or destroys this alien antigen? And there's different ways. And again, we'll just talk about one specific one, which is B cells and the production of antibodies. All right, let's get to specifically what's happening with B cells. So the clonal selection theory, specifically as it relates to B cells, to B lymphocytes. And here's how most of us like to represent this. These are B lymphocytes. Each one of these is a B lymphocyte. And on the surface of the B lymphocyte, there are receptors. Those receptors are actually a type of antibody that the B cells produce. These are naive B cells, which means that these B cells have never, ever, ever seen the alien that they exist to go after. In other words, they've never seen their particular antigen. And I've drawn 4 of these B lymphocytes, but keep in mind that in your body, populating your lymphatic tissues, your lymph nodes, your spleen, your tonsils, there are billions of B cells, billions of these things. I'm not going to draw billions. I only have room to draw 4. Again, each one of these is different. When they developed in the bone marrow, the particular type of alien antigen that their particular receptor or their particular antibody would bind to and therefore attack, that was determined in their early development, again, through a rearrangement of the gene that they have for that receptor or that antibody. So 4 different B cells, each one of them is going to attack a different alien antigen. Now let's introduce antigens. So let's say you either got an immunization, you got a shot, or you got infected with a virus or a bacteria. Within a lymph node or other places, you will find bits of that alien floating around, bits of this alien antigen floating around. And let's say that this alien antigen just happens to be the right antigen to bind to this particular B cell's receptor. What happens then is these B cell receptors get together, and they usually don't get together. They float around on the cell's membrane. But the presence of this alien antigen causes these antibodies, these receptors on the surface of the B cell, which again is a type of antibody, it causes them to come together, and that sends a signal inside of this B cell saying basically, hey, this is our alien. This is the antigen that we were born to go kill. That begins activation, but only for this particular B cell. And again, that's the key to, or the beginning of, clonal selection. We're selecting for that particular B cell that is going to go kill the alien, or that's going to help go kill the alien later. With that said, I can get rid of these other guys. These other B cells are important, but they will be important for other aliens. We've selected this one B cell because it happens to want to go after that particular alien. All right, the next step. This B cell will phagocytize that antigen. It'll bring that antigen inside of its own cell, and it will display that antigen on what's called an MHC2. It's performing what we call antigen presentation, and it's presenting that antigen to T helper cells. And what it's basically asking is this: hello, T helper cell. I am a B cell. I'm a B lymphocyte, and I just found this antigen, and I really want to produce antibodies against it to kill it. But I want you to confirm that this is an alien antigen. Do you recognize this antigen? And by the way, in immunological terms, if an immune system cell recognizes something, that means it wants to help kill it. The B cell will show that alien antigen to a bunch of different helper T cells, because helper T cells are generated in the exact same way that we talked about earlier in terms of it's random in terms of what type of alien antigen they're going to be raised up against that they will go after. If this B cell happens to come across the right helper T cell, and that helper T cell says, hey, yeah, I recognize that, that's alien, go kill it. That starts the actual mitosis process and the full activation of this B cell. And by the way, this step, the importance of this step is it's checking to make sure that this B cell is going to attack an alien. It's not going to attack self. This is just a double check saying, hey, I think this is an alien. Let's make sure it's an alien before I go attack it. This helps to prevent autoimmune disorders. It helps to prevent your own body's immune system from attacking itself. This double check doesn't always happen, by the way, but I do want my students to know about it because of its importance in preventing autoimmune disorders. So again, the B cell was selected for because it came across its alien antigen. It internalized that antigen, displayed it on an MHC2, got confirmation from a helper T cell that said, yes, that indeed is an alien, go ahead and go kill it. And then what happens is the mitosis phase. Whoops, not mitosis. So mitosis happens, and we end up producing lots and lots of B cells. We go through a few turns of mitosis. I'm going to run out of room on my board, so imagine a few steps, and we end up producing lots and lots of B cells. And I should draw some of these B cells differently, so let me go back and do that. Because when this process happens, when the mitosis process happens, there are two different cell types that get produced. And again, I am limited by space. I can't draw millions, but you can imagine this one B cell that happens to that wants to attack that alien antigen. It divides multiple times and you get hundreds of thousands, maybe millions of B cells, and all of these B cells will want to go kill the same alien. All right, the types of cells that are produced by this cell division, these guys over here behave just like the original mother cell. They are ready if more of this alien antigen comes along, and they can become activated and go through the same process. So we call these B memory cells. And the importance of that is these B memory cells will last for years and years. And instead of having just one or two original cells that could recognize the alien antigen, we have at least thousands now that can recognize the alien. So you can imagine all of this can happen a lot faster, and we can ramp up the response faster when we have these memory cells. And again, they last for years. So this gives us immunological memory. We'll be able to kill the alien later. These cells are called B plasma cells. And these cells, they don't last very long. They only last a few days. Sorry, let me turn this off. They only last a few days because they literally work themselves to death, producing antibodies. And those antibodies leave the cell. I'm going to draw them very simply as these little kind of Y shapes as I've been drawing them so far. But these B plasma cells produce literally millions of particles of antibody. And initially, if we've never seen the alien before, these antibodies come in a specific form, which is IgM antibodies. They actually look like little wagon wheels. There's 5 of these little antibody particles bound together. 2, 3, 4, 5. But I'm not going to take the time to draw a ton of these little wagon wheel looking things. Just know that we get millions, possibly billions of little particles of antibody now. Those antibodies now go out into the body's fluids everywhere. And they will bind to that alien antigen wherever they come across that alien antigen. And that has multiple effects on the alien antigen. If it's a virus, now that virus can't infect other cells because it's got antibodies clumped around it, making it incapable of making contact and infecting other cells. If it's a snake venom to have its toxic effects, it has to be able to make contact with the molecules that it makes contact with to do the damage. If the snake venom molecules are coated with antibodies, they can't make that contact, they can't have their effects. And for any antigen, if it's coated with antibodies, other body defense cells like macrophages and neutrophils and such, they recognize, okay, I don't know what that alien is, but since it's bound by lots of antibodies, that's definitely an alien and it must be destroyed. So phagocytosis. That part of what antibodies do, by the way, is called tagging for destruction. We tag that alien antigen for destruction by other phagocytic cells, like, again, macrophages, that's the major one, but also neutrophils. So that's clonal selection theory. Again, remember we started out with lots of B cells, lots of different B cells. Only one of them was able to recognize the alien antigen represented by the red dot. So that one was selected for. The clonal part, this cell is cloned. It goes through mitosis, which is a form of cloning. We're making identical copies of this original B cell. And the clones that are produced come in 2 cellular varieties, the memory B cells, and those memory B cells, again, last for years and years and years. They can ramp up an immune response similar to the initial immune response, but now, since there's so many more B cells, we can do this faster. And then the other type, again, are plasma B cells. And those plasma B cells, again, they work themselves to death, producing billions of particles of antibody that will then go out and bind to that alien antigen and give it a bad day, whether that bad day is inactivating the alien antigen so it can't have its negative effects, or simply tagging that alien antigen for destruction. I guess another thing that antibodies do when they bind to alien antigen, notice that at its bare bones basics, antibodies have this Y shape. And there are 2 binding sites for aliens on even this basic Y shape. So what that means is this antibody can bind to alien antigen on both of its binding sites. Another antibody could bind to the same alien antigen, which can bind to another alien antigen, which can bind to another antibody. What you end up with is a clumping up of the alien antigen. If the alien is a cell, this is agglutination of that alien. It clumps the alien cells together. If it's a virus, then it's precipitation of this alien virus. And the helpful thing for this for the immune system, if this was a tiny virus, now we've clumped up all those little viruses into one nice ball. for a macrophage to engulf and destroy. So it mops up the alien antigen and clumps it up, making it easier for the body's defenses to get rid of that alien. And again, the wondrous thing about clonal selection theory is it explains why it is that you never catch the same virus twice. Usually, you will catch that particular virus, your body will defend itself against it, and you'll never catch that virus again. And it also explains why immunizations work. When we give you an immunization, we're injecting you or delivering in another way an antigen. Usually it's not the actual alien itself, like when I spoke about smallpox earlier. We no longer, when we do immunize against smallpox now, we don't give people the actual smallpox virus. We give them what's called cowpox, and we give it in the arm, and it does cause a little local infection. But now the person's body knows how to defend against actual smallpox. The other immunizations we can give for measles, mumps, rubella, tetanus, diphtheria, all of those immunizations work because they trigger this clonal selection of not only B cells, but remember that T helper cells, T killer cells, the other types of immune system cells, they go through a similar clonal selection process, including helping to make sure we don't attack self. And now, after the immunization, we'll have B cells that can go and attack the alien, we'll have T helper cells that can go and attack the alien, and we'll have T killer cells that can go and attack the alien if needed. So the clonal selection theory, again, helps us understand how it is that we can have long-term immunity against something. And it tells us how it is that our body responds to defend against the original or the initial infection as well. So that's clonal selection theory. Let's do a little quiz time just for fun. Taking away the helping words. So clonal selection theory in a nutshell, why should we care? And I hope your answer came something along the lines of, it helps us understand how our immune system can defend against something and not have to fight off a true infection ever again. You might catch the virus again, but your immune system takes care of it before you get sick. Kind of cool. So that's an example of why we care. In this initial step up here, whoops, I'm going to try to draw back the other B cells that I started out with. And maybe I'll draw in these little red dots again. Do you remember what the red dots were supposed to be? And here's a little curveball for you. We're inside the body somewhere. Where in the body are we likely to be? Do you know? So to the first question, what are the little red dots supposed to be? They are supposed to be some kind of alien antigen for some kind of virus or bacteria or something that's trying to cause an infection. And in terms of where are we supposed to be, usually when students are studying this, they're studying this along with studying the lymphatic system. Most of these lymphocytes, B lymphocytes and T lymphocytes, they live in the lymphatic tissues. So lymph nodes in your neck or under your arms or in your lungs or in your gut. So inside of lymph nodes, but also possibly inside of the tonsils, the spleen, or other lymphatic tissues. So that's where we probably are in the body, is inside of some lymphatic organ. What's going on in this first step up here? Why did I draw these 4 lymphocytes, and what do they really represent? And what's happening? And for that, I drew 4 of them because I didn't have room to draw billions. There are actually billions of different B cells inside of your lymphatic tissues. And in terms of what's happening here, what I'm trying to show happening here, out of the billions of B cells, one or just a few happen to recognize that alien antigen represented by the red dot. Those that happen to recognize that alien antigen, they are selected for. That's the selection. And then what happens next? What's going on between these two cells? And what's happening here, this is that same B cell. It has engulfed an antigen, this alien antigen, and it's displaying it on what's called an MHC2. MHC, by the way, stands for Major Histocompatibility Complex. It's a protein that cells use to display antigen. So the B cell is using an MHC2 to display that alien antigen, and it's checking with helper T cells, and it's trying to confirm that this is indeed an alien antigen. In a way, the B cell is saying, hey, this is my alien. I really want to go kill this. Is that okay? Can we confirm that this is indeed an alien and that I'm not trying to kill self? By the way, if that confirmation, if this cell is looking for that confirmation and it never comes, this cell will eventually kill itself to help prevent it from attacking you. But let's say the helper T cell, one of the helper T cells said, yes, that is an alien, go. You can go ahead and go after it. What happens next? So what's happening between this cell and these cells down here? This next step, and it's more than one step, I could only draw one, but you can imagine this cell divides into 2 cells, which divide into 4 cells, divides into 8 cells, and so on, and we end up with hundreds of thousands or millions of B cells that will attack the same alien antigen. That is the cloning part of clonal selection. We've made multiple copies of this original cell that can attack the alien. There are 2 different types of cells represented down here. What are those 2 types of cells? And those 2 types are, these 2 are supposed to be memory cells, and again, there's not just 2 of them, there's thousands or millions of them now. Memory cells that can react to the alien years and years from now. But also they could react right now if the infection continued, they got exposed. They could react right now if the infection continued, they could actually ramp up the response and go through this process. But I like to think of it in terms of what's happening years and years down the road. So memory B cells, they can react to the alien antigen if it comes back again years down the road. And this is the really critical thing for the initial story, which is, why is it that we only get infected by this virus once and then we don't get infected again? We can be exposed again and we don't actually get an infection again. These memory cells are what that's all about. This is immunological memory. The memory cells that get produced by the clonal selection theory, those memory cells are what give us immunological memory, prevent us from being infected again. What about these cells? What are they? And those cells are supposed to represent B plasma cells or plasma B cells. By the way, the extra stuff I drew inside of them, that's supposed to be rough endoplasmic reticulum for you basic bio geeks. Remember that rough endoplasmic reticulum, lots of protein production and protein processing. In this instance, protein production and processing for making tons and tons of antibodies. Again, these plasma B cells, they're short-lived. They only live a few days. They work themselves to death, producing billions of particles of antibodies. And those antibodies then, of course, do what? What do the antibodies do? The antibodies, of course, give the alien a bad day, and initially they do that just by binding to the alien. They bind to that alien antigen, and depending on the antigen, then the effects they can vary a bit, but some of them, again, are simply tagging that alien for destruction. Now the other white blood cells, macrophages, neutrophils, they know that whatever's clumped in those antibodies, we got to destroy. So it's tagging the alien for destruction. Inactivating the alien antigen. If it's a virus, now the virus can infect things if it's bound by antibody. If it's snake venom, it can't have its effects because it's bound by the antibodies. So most snake venom, by the way, they're enzymes of some type. They do some kind of chemistry that's damaging. And if those antibodies are bound to the, if the antibodies are bound to the snake venom catalysts, what are they? Biological catalysts, enzymes. My gosh, I'm tripping over enzymes. Anyway, if the antibodies are bound to those enzymes that are the snake venom, the chemistry, whatever the bad chemistry is that they do, they can't do it. So inactivation is the point. Sorry, I went too deep on that. So tagging for destruction. Causing the alien antigen to be all clumped up to allow us to clear it better. Those are some of the examples of what the antibody does to give the alien a bad day. And it all just starts with attaching to that alien. Once the antibody's attached to it, that causes all kinds of issues. Oh, one that I forgot to even mention earlier, the activation of what are called complement proteins. If that alien happens to be a cell, a large cell, if that cell has antibodies bound to it, then there are these proteins in your bloodstream that get activated, and those proteins are called complement proteins. Those complement proteins will make holes in that cell that the antibodies are bound to. That puts lots of little holes in that cell, and that cell is going to be killed as a result. So anyway, that's clonal selection theory. And again, broadly, the most important thing that it explains, I think, is what we started out with, which is, again, even our ancients noticed that if you get infected with a specific infection, you get sick from it, but then you get better, assuming you survive, and you can't get infected again. This is why you can't get affected again, why you cannot get infected again, and it's because you end up with these memory cells. You end up with immunological memory. So the alien might get into your body again, but immediately the immune system cells produced by the clonal selection theory, those memory cells produced, they're going to ramp up and kill that alien before it even causes you an infection in most cases. And that's clonal selection theory. As always, if there's any questions or comments, please leave them down below. And thank you once again for watching.